Again, thank you for joining us for our Junior RNA Scientists Representative Town Hall. We're really excited to be here to tell you what our role is as Junior RNA Scientists, who we are as scientists, and really to get your feedback on how to improve our advocacy for Junior RNA Scientists and to answer any questions that you may have. Now, how our sort of committee structure works is each year we select 2 members of our Junior RNA Scientist Representative Group who will volunteer for a 2-year commitment. I'm Cameron Barry. You'll hear I'll introduce myself a little more in detail in a second, but along with Morgan Lucas, we're the returning representatives. So we served last year as RNA representatives. And we're excited to also have Samuel Ogunsola and Christian Ramirez, who joined the team in January. And so the structure for this town hall is that we will introduce ourselves and who we are as RNA scientists, our connection to RNA. We'll then dive into a bit of our roles, some of the activities that we've organized in the past as Junior RNA Scientist Representatives for the RNA Society. And then we'll really end with focusing on answering questions, some of which who have been pre-submitted, and also some of which we're hoping that you'll submit through the Zoom Q&A. And I also want to give a shout out to one of our great PI supporters, Carol Lutz. Carol, along with Pat Baldridge, really helped support us as Junior RNA Scientists. And so with the structure, let's start off with Morgan. Morgan, if you can introduce yourself to the group.
Sure. So my name is Morgan Lucas, and I'm an industry scientist working in genetic diagnostics in Germany. Why I'm fascinated about RNA and work in RNA research is because I think it's just one of those key functional layers of gene expression. You know, DNA stores information, but RNA determines how that information is read, right? And it's incredibly dynamic, responsive to the environment. And there are so many different types of RNA classes and mechanisms that regulate them. And I think most importantly, we just don't know even 1% of what there is out there about RNA. So my work in RNA now is quite different from my basic research during my PhD. So I'm working on using RNA as a functional readout of DNA variants in diagnostics. So in genetic testing, we often identify through sequencing DNA variants that we cannot use diagnostically because we don't know whether they have a real biological effect, if they're pathogenic or benign. So RNA analysis can help us reveal those. So my focus is on implementing RNA approaches into the clinic. And the reason that I joined so I've been a member of the RNA Society for a long time, all through my PhD and now is my career as an industry scientist. But the reason that I joined as a rep is because I really wanted to give back to this community that's been really supportive through my research career. It's a really warm and generous community, and that culture can continue through active involvement of its members. And also by joining a committee, you just learn a lot about the system that you're working in and how it functions. So I find that perspective really valuable.
Awesome. Thanks so much, Morgan. Samuel, if you want to follow up with your intro.
Thank you, Cameron, and hi, everyone. My name is Samuel Ogunsola. I'm a PhD candidate at the University of Manitoba here in Winnipeg. So, so why did I join the RNA Society, the Junior Scientist role? Last year, I was privileged to attend the RNA conference in San Diego, and it was a great experience getting to meet other young researchers like me, getting to learn about what they're doing and also how they are making impact through RNA. So that's kind of create an interest in me to also contribute and give back to the community. And besides that, being a Nigerian and African studying RNA, one thing is this underrepresentation of Africans doing RNA research. And even those that are doing, they are not probably well known. So part of my goal to as part of the junior scientists is to see how we can, include more of these people in the community and bring them into the society and make them more active. So what am I working on? I study alternative splicing. So I study a particular a set of group of elements located in the introns. These elements, we notice that they are enriched in the genome. However, we're yet to fully understand their functions. So in my PhD so far, I've been trying to understand what they do, and we've been able to publish some interesting findings and also get to understand them better. And this connects to my interest in RNA. One thing as a young scientist, one thing that really impressed me a lot about RNA research is the fact that how RNA, contribute to our complexity as higher organisms, right? Imagine a single gene and then, making thousands of mRNAs, making different proteins. That's fascinating, right? So that kind of bring an explanation to many of the things we see in our world today and what differentiates us from other organisms. So getting to understand this is really amazing. And that's why I study, love studying RNA. So it's good to be here and really looking forward to an amazing conversation. Thank you.
Awesome. Thanks so much, Samuel. Christian, do you want to give us an introduction, please?
Hi, I'm Cristian Ramirez. I'm a PhD candidate at the University of Trento in Italy, which is basically in the north of Italy between the Alps. So I'm interested in RNA basically during my short career for now. I've been fascinated by how RNA is very important in a lot of cellular processes, and we are still discovering a lot about it. And it seems that the interplay between a lot of different factors really is implicated in many processes. And in fact, what I'm working on is how RNA modifications affect the RNA metabolism and how they are implicated in diseases through their interplay with RNA editing, RNA structure, and protein RNA binding. And as a bioinformatician, I'm basically analyzing all of these different omics, trying to figure out what is going on. And why I'm interested in why I was interested in joining as a junior RNA representative. It's basically because, I discovered the RNA Society a couple of years ago when I went to the Edinburgh conference, and it really was a great experience for me. Also discovering, getting to be kind of part of this big community, and all of the activities are organized by the Junior RNA Scientists. I thought they were wonderful because they were a great way to involve younger scientists and to create a community that makes you feel a bit more connected in a huge community. So I really wanted to try to give my help in making in continue improving this.
Wonderful. Thanks so much, Christian. And so I guess I'll introduce myself in a little more detail. So I'm Cameron Barry. I'm a postdoc at the Stowers Institute for Medical Research, which is actually located right in the middle of the United States in Kansas City, Missouri. And I actually came through my interest in RNA through a slightly roundabout way. And so I started my PhD in developmental biology, and I got really fascinated actually with this tissue-specific alternative polyadenylation. And so what does that mean? It means essentially you can have the same gene can have its RNA processed in different tissues to lead to different lengths of 3' UTRs and different lengths of RNAs. And something that's actually quite conserved across many different animals is that in the brain, RNAs are expressed with really long 3' UTRs, and in the germline, they're expressed with really short 3' UTRs. And so for my PhD, I ended up figuring out in the Drosophila male germline how these 3' UTRs get so short. And we actually found out that in the stem cells, they're produced long, and then they're actually differentially processed as those cells differentiate to get shorter. And we did a little bit of work to show how that can impact the translation of those RNAs. And through all that work, it really got me into the 3' UTR field. I really was kind of inspired by, I think, how much unknown there is in this field. And so I started my postdoc with Dr. Adiel Bazzini here at the Stowers Institute. who had actually just discovered quite non-canonically that there can actually be translation in 3' UTRs. And so we call those downstream ORFs. You may have heard of upstream ORFs in 5' UTRs, but there's also small translated regions in 3' UTRs. And so that kind of blew my mind. And so I've been really working on how do these regions in 3' UTRs get translated, and do they have regulatory roles for gene expression? And so, yeah, so I've really been on this 3' UTR kick for a little while now. But I really chose to become a junior RNA scientist representative because I really wanted to empower junior RNA scientists like I was, and like backgrounds like where I came from as a first generation college graduate. When you start university, I had no idea how scientific research worked or how academia worked. And so I really think that finding ways to build community, kind of like Christian said, and organize events that really help to demystify academia and make it sort of more welcoming, and especially spread the good word about RNA and all the really cool things to do there. And so those are sort of the angle that I have on sort of the things that I've been contributing to as in the role as an RNA scientist, and the things that I hope to keep contributing on. And so with that, that'll be a sort of, yeah. So now that we've introduced ourselves, we can hop into some of the different activities that we've actually organized in the past few years. And Morgan, do you want to kick us off with that?
Yeah, so last March, a former junior rep, Belen, moderated an online career panel. It was called Alternatives, with the RNA part in capitals, and it was celebrating women's careers journeys in science. This was for International Women's Month, where we heard from highlighted successful women in science with diverse career paths, and that was really interesting.
Yes, that was a lot of fun. Yeah, I helped moderate that, and yeah, I'm really excited for some hopefully future panels that we'll end up organizing and moderating. One other sort of activity that we've done, this is actually, we're really hoping to continue this as sort of an annual activity, is on RNA Day. So AUG in August, right? We've been hosting this baking and art competition. And I really like this because it really highlights and sort of allows a venue for artistic expression through our scientific community. I think sometimes, we kind of think these are two different things, but I think we all know people who are really able to really excel in both of these angles. And I really enjoyed it actually because my sister's a pastry chef, and she had been making all these macarons, and so it inspired me to sort of get her recipe working in my hands. And so last year I sort of made macarons in different shapes and sort of organized them as sort of translating ribosomes as, macromolecular machines. So really, if this sounds exciting to you, keep an eye out. You should hear from us around July, hopefully in July for the next one. And last year we actually were sponsored by GenScript, and there were some really cool prizes. So if that's up your alley too, keep an eye out. And then, yeah, does someone want to talk about sort of our role that we play at the RNA Society, the annual RNA Society meeting?
Yeah, I can take this one. So obviously my experience is for I'll restart. So generally, there's three main events that are organized by the Junior RNA Scientists at the RNA Society meeting. One is a career panel that can be of different sorts. For example, when I attended, it was a panel where there were people who, after their PhD, went to continued in the academia path, and others that went to industry. And so they both talked about their experiences, so that junior scientists could have a different perspectives on the pros and cons of choosing either path. But in different years, this topic changes, so that it always varies. And then there's more of an informal event that can be, for example, an excursion in the city, for example, even just like a pub crawl, which doesn't seem that scientific, but at the same time, I think it's a great way to build connections between junior scientists, so that you feel more free to talk and connect with other people that are more or less at your level. And so I think it's a great way to get to know new people that are from all over the world in a more informal setting. And finally, if I am not mistaken, there's also an event right before the conference that a couple years ago was a communication event with an expert on communication. And so we will also organize something of that sort.
Yes, exactly. Thanks so much, Christian. Yeah, and the truth is, every year we really have a different set of events. We really like to sort of adapt and give the community what they need, and that's one of the reasons that we're here today, is to really see sort of what suggestions or to listen to the questions you have for us, so we can learn about and sort of organize some future events. And so we're really exciting. You'll definitely hear from us for some upcoming events for this year. But now we can transition into sort of answering some pre-submitted questions. So now we're kind of going into the Q&A portion of this. And we had previously sent out a survey, and we got some nice responses to that. So we're going to go through some of the questions that were submitted there. And then just as a reminder, if you have any questions right now, feel free to submit them to the Q&A, and Carol will help us address those afterwards. But yeah, so let's address some of the pre-submitted questions. We had first, the question got submitted was who counts? Like sort of a nice question, like who is a junior RNA scientist? And does anyone want to tackle that question?
Maybe I can answer this. So because I just applied last year, so I think I got some details about who is a junior RNA scientist. So this includes undergraduates, master students, PhDs, postdocs, and early PIs in the RNA Society. So everyone count as junior RNA scientist. So in case if you find yourself in this category, or you have a student in your lab that you feel should be part of this community, it's a good opportunity for them to join us and also to follow all our programs. And then you can also use the opportunity to network and connect with other young RNA scientists across the world. Yeah.
Awesome. Thanks, Samuel. Yeah, so we'll move on to another question that was asked to us. The question was, what is something you enjoy and something that you have learned while being an RNA Society junior rep? And the truth is, for me, the thing that I have enjoyed most, I think, is the camaraderie between our group here. Maybe I'm a bit biased. I really do think we recruit people who are really passionate about investing their energy and giving back to the RNA community. And I know that for me is very energizing. And I think one thing that I learned, I think in my training, I've developed sort of a comfort with giving scientific talks in front of people, but I think I've been a little bit more intimidated taking on different roles beyond that. And then last year, I was able to moderate a few different panels, and here we are leading a town hall. I really think that's really helping me to sort of grow my skill set, and things that I've really learned from that is preparation goes a pretty long way. With just feeling comfortable and sort of doing something new. And sometimes you just have to just do it, right? Put yourself into these opportunities. And for me, that's when I found sort of I've learned the most, when you put yourself in a new experience. So yeah. Morgan, do you have a question? I think the question was
yeah, so we had one that was, what soft skills have you learned or improved or hope for the new reps to learn or improve during time as an RNA Society rep, as an RNA Society junior rep, sorry. So for me, it's hard to choose. There's so many, but I would say communication and collaboration. It's just a great opportunity that you're able to work with so many different career stages, institutes, people from different backgrounds, different countries, and you have to learn to communicate clearly and take into everyone's perspectives. And that's really amazing. I've learned a lot through that. And I think for new representatives, it's a really great opportunity to build confidence in networking and collaborative engagement, and also community leadership. I think this is a really nice community to try and build those skills as a junior rep.
Awesome. Thanks, Morgan. And then I'll take our next submitted question. The question was, how much time do you commit in average to this role? And how do you manage that with research? Right. And the short answer is, right, it varies month to month, but I would say around, 5 to 25 hours a month. Depending on the month. What's really nice, right, is we have a whole team here where we can work together and share responsibilities. Right, the four of us, and then our faculty mentors, Carol and Pat, really help sort of split the burden. But some months you're organizing a few different things, and as a postdoc who's looking to go on the job market in the next couple of years, is definitely balancing a lot of priorities. And one thing actually, when you're in these situations, you find the solutions. And one really cool solution actually came from Morgan, who really introduced us to these project management software. We actually use something called Trello. I'm sure there are other ones. But I've really found that really helps break up some of the big tasks we do into smaller tasks. And so I don't have to give up a whole day or half day in the lab. I can find some time here and there. And if we organize that Trello really well, I can knock out some smaller tasks. In shorter time periods. And actually, interestingly, I've introduced this to my lab and my boss, and it's also like great ways to just organize your own schedule and group projects within the lab. So yeah, I think that's how I manage. Yeah, and it sounds great on paper, but it does take some juggling for sure. Perfect. And then,
Sorry. Another question that we had was, how can I become a junior scientist representative? And I'm glad that someone asked, because I hope that you will apply, whoever you are, in the next call. So as Cameron mentioned, we usually have 4 representatives, and we serve for 2 years each, and they're staggered by 2 terms. So we always, in our annual call, have 2 new representatives to recruit. And typically this call opens in September or October, and it's announced by our social media platform. So use X, Blue Sky, and we have a LinkedIn group. If you're not in there already, you have to request it. And then we also have a new WhatsApp community group that I think Christian will show us. And it's also put in the RNA Society mailing list. So you should hear about it one way or another. And in this call, we ask applicants to submit a 2-page CV and a half-page motivation statement. And we really ask that the applicants focus on their research experience, education, leadership and service, and outreach activities. Then we go through those applications. The interviews take place the following month, so that's around October, November. And it's a 30-minute interview. We give the questions in advance. It's the same every year, actually. And then we select 2 candidates. And then we also talk to their PIs before the official offer to make sure that the PI or supervisor, whoever it is, one, that they support the participation, and also that they're willing to cover the costs of the travel to the conference. So as a junior rep, we attend the RNA Society conference during our 2 terms, and this is covered by the RNA Society. So the registration for the conference and the hotel, and then your supervisor agrees to cover the costs of the travel. And Yeah, in this process, we really make sure we can have diversity across gender, ethnicity, geography, and career stage. So we like to have PhD and postdoc levels covered. Yeah, and if you know someone that should apply, especially if you're a PI or a senior scientist, please encourage students to apply, because I think not enough junior scientists take this opportunity or read their emails. So please, yeah, if you know someone, tell them to take this chance.
Awesome. Thank you, Morgan. We had another submitted question that says, how can I stay up to date with the activities organized by the RNA Junior Scientist Representatives?
I can take this one. So as you can see in the chat, there are three main ways in which you can stay up to date. Two more, I would say, classical ways are through social media, so X, Twitter, or Blue Sky, where we will post all of the new events, our questions for you. You can contact us for questions for us. Any way in which we can get more involved with the junior RNA community, and you can get more involved with us. But at the same time, we are setting up a WhatsApp community for which I've sent the invite on the chat. And in this community, there will be an announcement board where you will see all of the new events, just like on social media. There will be a general chat, and then there will be a group for the specific group for the conference, in which there will be more conference related information, and in which the junior scientists at the conference can connect with one another. So yes, we're still this is the first time we are using an actual WhatsApp community, so we're still figuring out how to work well with it, but we think it could be a great way to get more involved with all of you.
Yeah, thank you so much, Christian. Yeah, we're really excited for these new ways to build community. It really is a goal of ours this year to really build out our junior RNA scientists community and really get more engagement. So if you have any suggestions for us on that, we are 100% open. Speaking of suggestions, we also got a suggestion submitted, as well as some questions. And the suggestion says, I'd like if the junior RNA scientists could hold more workshops on developing skills to effectively mentor others, improve my ability to advocate for my career goals, and other interpersonal skills that could help my career path. I don't know. Does anyone have any comment on that?
Yeah, I really like this suggestion. I think they're really important skills to develop, and developing skills to mentor others and to advocate for your own career goals are actually not suggestions I heard from the community in my time as a rep. So I think that's really a great idea, and I think we can take this to the RNA Society mentoring initiative. I can add it to the Trello to see if there's an opportunity to organize something between them and the junior reps. But I also do recommend that anyone, junior scientists, that they sign up for the RNA Society mentoring initiative. So this is a one-on-one mentoring. You get set up with someone based on what you're looking for, if it's career advice or transitioning to a new, to industry or to academia. So I think this is a really great way to do that. I will put the link for the initiative in the website, in the chat. I did this myself, and it was great. I did this last year, and the funny thing for me, this is such a good opportunity, but there's always more mentors than mentees. So if you want to be mentored, sign up. And I think another piece of advice is just to ask PIs and senior scientists, and also joining committees like this. You get to work with a lot of senior scientists that mentor you. Like, for example, we have 2 faculty advisors, one who's here, Carol, and we get a lot of advice, not just being junior scientists, but on our careers and applications and things like this. So in those committees, you're meeting people that are really engaged with junior scientists. So they're really the best people you can get mentoring from, I think. But I really love this question, and I think we'll definitely take this back to the RNA Society mentoring team to see if we can do something there.
Awesome. Thanks so much, Morgan. Yeah, I really like that perspective. And then we had this our last submitted question. It's relatively open-ended, but the question is, how can we support our junior RNA scientist reps?
Okay, maybe I can go first on this. I think one of the first thing is to follow us on all our social media platforms and also join our WhatsApp community, because that will make the work easier for us. We can pass across information easier. And the other thing is try as much as possible to join all our programs and event organizings. These are very strategic programs, mainly to help us and also to make the community grow together. So please try as much as possible to do that. And if you're a PI or a senior scientist, please, you can support us by encouraging your students to join this community, so they can get to know that they are not the only one working on RNA. There are actually a lot of people across the world, young people also studying, doing RNA research. And just to add to, the other way you can also support is by sharing some of the informations we've shared here with other people. So maybe there are some people that couldn't make this meeting and you know that they are junior RNA scientists. So you can share the community information with them so they can join. And lastly, the RNA conference is coming soon. So one of the ways you can support is by attending all the programs we're putting together. So through that, we can get to network, know you more, and also see how you can support us more in the community. So if there's any more, I think others can add.
Wonderful. No, I think you hit a good amount of the points for sure. And yeah, we're really excited for the RNA Society conference coming up in Montreal. Okay, that is all of the pre-submitted questions. So we can move on to see if there's any questions in the Q&A. If you got a burning question, I would say type it up, as quickly as you can and hit submit.
So I just wanted to suggest that you talk a little bit about some of the things that you have planned for the RNA Society meeting in Montreal.
Yes, yes. So we are really excited. So we've been collaborating with the organizers for the Montreal RNA meeting, and we don't have everything completely figured out yet, but we have some really exciting plans for a kickoff to like Christian had said. Sometimes we do these sort of excursions, whether we do sort of a pub crawl or we do a hike or we just sort of hang out. We're hoping that before we go out on this, we'll actually have this sort of in the park sort of fun, perhaps like a barbecue-like situation. Don't hold me too much to all of it, but it looks like these plans are sort of are in the works, and I'm really excited to see.
And the panel?
Yeah, maybe you can introduce, because Cameron and Christian, you're moderating a panel this year in the RNA Society conference, right?
Exactly. Yes. So the panel is actually going to be called RNAi. We won't specifically be talking about small RNA silencing mechanisms. We'll be talking about the role that artificial intelligence, large language models, deep learning models can actually play, in scientific research, and how that affects our day-to-day work in the lab. And so we're really collecting a variety of really expert panelists that can speak on this from many different angles. And their perspective will be really exciting to hear. And of course, if you're there, we encourage you to attend our session. And also, of course, we'll have a Q&A, so you'll be able to ask any burning questions that you have for them. So yeah, we're really excited for that. I think, right, this is what a lot of people are thinking about. And so I think having that expertise all in one place will be really exciting.
And then following, I'm really excited for the panel. Yeah. And then following that, there's a networking reception that is open to the junior RNA scientists, and then following that, it will be the barbecue that Cameron mentioned, which looks like it's really going to happen, and it's going to be quite fun.
Yeah, we are super excited about that. I don't currently see any questions in the chat, but I do definitely want to let everyone know that you can reach out to us at any time. So we have an email address, junior_scientists@rnasociety.org. You can reach out to any of our social media platforms. You can DM us. If you find our email, however you can find a way to contact us, we would love to hear from you on suggestions, or if you have any questions, because we really are here trying to build a stronger community for junior RNA scientists. But yeah, with that, I just really want to thank everyone for showing up, and I hope you got to learn a little bit about us, a little bit about our role that we play as junior RNA scientist representatives, and I hope you'll feel more comfortable reaching out to us in the future with any questions you have.
Yeah. And just come up to us at the conference, please. We'll be
Exactly.
wearing big green pins, so you'll be able to find us.
Well, thanks so much, you all. Have a great rest of your day.
Thanks.